I gave you this study guide on your notes for this because this happens to be almost my favorite topic. And consequently, I love this and I want you to know it. And students have a hard time keeping up with what's going on. There's going to be 2 lectures on signaling to help you stay on track with what we're talking about. So this is talking about cell-cell communication, right? We communicate with one another pretty easily, right? How do we communicate with one another? Is it Leela or Lila? Layla. Layla. Oh, great. Leela, Layla, Lila, Lola, Lula. We'll use them all. How do we communicate with one another? We talk. Michael? Look, they're looking at her like, she spells it Michael. It's driving me insane. Her name is Michelle. Oh. You're just going to ignore me from now on, aren't you?
Yeah.
Okay.
Just like yesterday.
Just like yesterday. All right, so Michelle, how do we communicate?
You go.
I'm trying.
Using our language.
What? How else do we communicate other than talking?
With our hands.
With our hands.
Our mouth.
Eyes.
Eyes. What else?
Body language.
Body language.
Sign language.
Sign language. What other forms of communication can you use in
Text
today's?
messaging.
Facial expression.
Text messaging. What did you say? Computers. Email. How did they do it in the old days? Letters.
Morse code.
Morse code. What's this? Smoke signals.
Pigeons. There you go.
Pigeons. There's lots of ways to communicate. Lots and lots of different ways to communicate. And you're going to see that your cells also have a variety of ways to communicate from one cell to another. So one of the things we're going to talk about are what are the signals that go from one cell to another. But also, how do you receive a signal? Right? How do you receive my signals? Michael, Michelle doesn't receive my signals. She's blocking my signals, right? Because she doesn't want to communicate with me until I get her name correct, which I don't really blame you. You know, other people will text me back or email me back and say, what the fuck, right? What are you talking about? There's lots of different ways, right, to tell somebody you've received a signal. But once you receive that signal, right, like let's say I give Ben a signal. I slap him upside the head. You know, that's a signal. And he receives that signal in a particular way. What's the receptor that's receiving that signal?
Pain receptors.
It could be pain receptors, right? And then what's that going to do inside of Ben?
Turn on the fight or flight mechanism.
Ears.
Turn on the fight or flight? Really?
Adrenaline.
If I slap you upside the head,
Immediately, you might get flipped.
okay, I'm not going to do this. That's dangerous. Okay. Let me go pick on mine. Because I'm pretty sure she's not going to flip me. Okay. And if you tried, I think you would have a lot of trouble. I'm really heavy. So the intracellular things that happen are really important because they give you a function, right? So for Ben, it's either he's going to punch me back or he's going to get up and run away because he's so afraid of me. For Michelle, right, for her, she might say, oh, Dr. Stein is trying to teach me something. I'm going to think about that and process it and learn from it. I'm just saying. Okay? So all of these things are integrated together. So the extracellular signal has to be received. Once it's received, it has to be translated into another signal. It gets processed. And once it gets processed, then it has to go wherever it goes inside your cell to do whatever the function is. Some of the things that are most commonly used, right? Cell surface receptors are the things that receive a signal. And then intracellularly, the things that we know function are changing the conformations of proteins. So what does a kinase do? It phosphorylates things. So if you activate a kinase inside of a cell, it's going to phosphorylate something. And once it phosphorylates that something can now be turned on or turned off. Okay, and what does a phosphatase do? It takes off a phosphate group, so that can either turn something on or turn something off. GTP binding proteins. So if you have a protein that's got a GTP bound, and you take that off, or you cause it to be hydrolyzed, that can turn it on or turn it off. So all of these things can turn on and turn off signals intracellularly. And ultimately, right, we're trying to do something. The cell is receiving a signal. What do cells receive signals? What do cells have to know? Do your cells have to know anything? What do they communicate? Who communicates with whom? Every cell communicates with every cell. Not really. Neurons communicate with what? Specifically like muscle cells. So, right, we talked about how muscle cells get activated. A motor neuron synapses with a muscle cell, and the muscle cell then does something. What's the signal that the neuron actually emits? What does it send out?
Action.
No, so an action potential is what's inside of a neuron. A neurotransmitter. So it's sending out a chemical signal. Neurotransmitter. What is that? It's just a protein. Serotonin, acetylcholine, epinephrine, right? Different neurotransmitters. How does that end up in you? Does that acetylcholine do anything as far as a muscle contraction? Right? The muscle contraction is regulated by, you know how it works, right? Calcium. So we have to get calcium, so that whole signaling pathway, that's what we're talking about here. Acetylcholine bound to the acetylcholine receptor, that caused an intracellular cascade that caused the calcium to be released from internal stores, the sarcoplasmic reticulum. That was through the opening of a channel, a calcium channel on the sarcoplasmic reticulum. So how does that all work and function? I like this slide because it's simple, all right? And simple sometimes is good, but you know this isn't going to be simple. So this is showing you, like, you could have a transmembrane receptor that binds a ligand that activates protein number 1, that activates protein number 2 in any multiple ways, that activates protein number 3. And ultimately downstream, you can get all sorts of things activated in a cell at the same time, right? You can change metabolism, right? So you can maybe stop doing glycolysis right now because you don't need to. Or you might want to ramp up glycolysis because you need more energy. Okay, you can turn on and turn off genes. So that means you're going to activate transcription factors. You can change the shape of the cell by changing cytoskeletal filaments. Right? You could even turn the cell to do apoptosis. So maybe the signal is, holy crap, we're infected by Ebola virus, kill, 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 meaning kill myself, because I don't want to spread the Ebola virus. So all of these are going to be downstream targets where the signal is. All right? Extracellularly, we're talking about a signal that's totally different than what this internal signal is. And, right, you have to, so when I give you an extracellular signal, right, that's an extracellular signal. That means you can interpret that in any way you want, right? It just depends. Like my friend Taka, who didn't know what I told him it means you're number 1. I did explain it after the fact, but, you know, because I didn't want him to go out and tell everybody they're number 1. Hey, you're number 1! But it depends on who you are, right? So Taka was all like, oh great, I'm number 1. And Nicole would be like, fuck you, I hate you, right? I mean, it's different responses depending on who's getting the signal. Different things, so we're going to talk about some gases that do signaling, all right? Proteins, peptides, nucleotides, there's all sorts of things that can be signals. And they're usually made by one cell and secreted by exocytosis, and then they can reach another cell. And that can either be they can reach a cell right next to them, or they can reach cells really far away. What other thing have we talked about that signals far away? What'd you say? The blood. Well, it's going to be carried in the bloodstream. So what molecules are secreted by one cell and act elsewhere? Hormones. Like insulin. Right? Insulin is made in your pancreas. Right? And where does it act? What cells does insulin work on? Fat and skeletal muscle. So that means, right, I have skeletal muscle in my toes. I got really strong toes. But I mean, that means that insulin has to be secreted from here and go all sorts of places. Those are far acting, and that has a name. Does anybody know what that name is? What kind of signaling? It's called paracrine. I think this, okay. Paracrine or autocrine, I think, is another one. This is saying short distance. Paracrine is short distance. I could have this wrong. Is this right? Short is autocrine. I know short is autocrine. I'll have to look this up. I'm pretty sure this is right. I've been teaching this for years. Just can't remember what the fuck I'm doing. Sorry. This is saying paracrine is short distance. So this cell is secreting something, and it's only working at short distances.
That's what Wikipedia says.
That's what Wikipedia says, short distance? Okay. Some of the cells actually have to be in contact with one another, and that's really short distance, right? That means, that's like the difference between killing somebody with a gun and killing somebody with a knife, right? I mean, a knife is up close and personal unless you're able to throw knives, right? This is, you actually have to have contact between the cells to get that signaling. Endocrine, shit. How could I not know that? I am so stupid. Endocrine, right? What's the endocrine system all about? Hormones. Dumbass. I'm a dumbass. So endocrine is far, okay? Far distance. And so if things are really close, you can signal really quickly. If things are very far, it takes a little longer. It doesn't really take that long. If you think about insulin, how long does it take for insulin to act?
Pretty quickly.
In your body? Well, it starts to be secreted, but it actually functions approximately 20 minutes later. All right? You eat, and to get the signals going and to get enough insulin in your body, it takes about 20 minutes to get it upregulated.
You mentioned autocrine, so I wanted to...
Autocrine
paracrine are like the same thing?
Autocrine is a cell that's secreting something, and it acts on itself. So you can actually...
Or cells of the same type?
No, autocrine is itself. Does it say the same type? Fine, the same type of cells. So, but think of this. So autocrine is activating yourself. You all know how to activate yourself, yes? Okay. You imagine the fact that you're activating yourself. As long as you had like 12 of you around you, you can do the same thing on all 12 of you. There you go. How's that? Bad. It's bad. All right, endocrine's far, blah, 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 takes longer. Neurons is far, and they're far because the axons can be really long. The best part of this is we teach this in a very, the only way to teach this is by minimizing it down, breaking it down to one thing at a time. But that's not how it happens at all. There's a billion signals happening all at the same time. So a cell can receive signals A, B, and C, and this cell, when it gets A, B, and C, it's survive. It's okay, do whatever you need to survive. Another cell, right, this is the same type of cell, if it gets A, B, and C plus D and E, that might mean, okay, not only survive, but now I want you to divide because I need more of you. Okay, here you have A, B, C, F, and G. Not only do I want you to survive, I want you to differentiate into a different kind of cell. Yes?
So
So the cell needs those to survive and the signals just to survive?
Yeah, cells need signals to survive. In a lab, if you just plate a handful of cells in a dish, they're really unhappy. They need neighbors, and those neighbors secrete things to help the other cells survive. It's a really bizarre thing. Yeah.
Do we know how any of these things happen, or we just know?
No, we know a lot about all of this, and that's what you're going to learn. You're going to love it, Michael.
What about single cell organisms?
Single cell organisms, right? They still like to have friends. They can survive for some time. They'll grow and divide and grow and divide, and they're never really single for very long, right? So sometimes, right, no signals means the cell's going to die, in particular in the immune system. So this is really the only place I know this from. In your immune system, cells that get made, they have to receive signals, and the signals come from cells that they're interacting with in the body. I'm not a single cell person. This is not who I am. So even though I work on bacteria, I don't know. Yeah.
What if they got signal A, C, and D, but not B? Would that also?
That's just going to be a different in a different way. And even if you have A, B, and C in this cell that said survive,
It could be.
in a different cell, that can mean die.
Okay.
Same signal, totally different response. Okay? So this is hard to understand. You're not really meant to grasp it all fully. But let's start talking about some of the signals. All right, if I go to the last slide, is it on? Does this really fuck everybody up? This isn't even the one. On the last slide on the second lecture, you're going to see a signaling cascade of, it's just a crazy picture of a billion things signaling. That's one receptor signaling. It does all these things, and then you have not just one receptor, you have multiples of that receptor, you have multiples of other receptors. So it all turns into this crazy mix mash of how does that work? How does your cell know what to do? We have no idea how the cell can do this. It's like a mini supercomputer. Each one of your cells is like a mini supercomputer. It integrates all of the signals and somehow makes it work. All right, so let's talk about some of these extracellular signals, the ligands that are going to work. Gases are really cool because they can just cross cell membranes. They just cross cell membranes without having to do anything. Okay? Nitric oxide is one of them. Name another gas that crosses the membrane that kills you. Carbon monoxide. Yay, that's a great one, yeah? We're like, woo. All right. So gases are nice because they are able to cross membranes, and other molecules, such as some hormones, actually can cross membranes without any problem because they're based on this steroid structure. Where do you remember this steroid structure from? Cholesterol. Right, it's part of your membrane, and it allows for some of these steroid hormones to cross membranes. What am I talking about? What hormones do you know of? Give me another hormone. Testosterone. What makes you a manly man? Right? What makes you a girly girl? Estrogen. Right? So these hormones can cross membranes and cause all sorts of downstream signaling. All right. We're going to talk about some signaling cascades, so I hope that's simpler. Let's start with nitric oxide. All right. Has anybody ever had nitric oxide? Where do you get nitric oxide from? Dentist. What do we call it? Laughing gas. Yay. It's really funny. I remember getting laughing gas. I remember they gave me the laughing gas first, and then they gave me a needle. I was like, ow! Somewhere in the back of I knew it hurt, but it wasn't that hurt. It didn't hurt that much. All right, but the nitric oxide, right, can freely get in. You breathe it in, and then it's taken into cells. And it's really interesting. It moves through gap junctions. So all cells have gap junctions. And it's really important for smooth muscle contraction and relaxation. Okay, what are we talking about when we're talking about smooth muscle? What has smooth muscle on it?
Stomach.
Your stomach. Your intestines. There's a really important one. Your heart. All right? All of your internal organs have some smooth muscle surrounding it. It's not skeletal muscle, it's smooth muscle. And so, one really interesting thing is that nitric oxide is actually made from arginine. What's arginine? Amino acid. Pretty sure those amino acids are going to keep coming back and biting you in the ass. Okay? On exams, maybe. So, oxide actually is made by an enzyme called nitric oxide synthase, or synthetase. NOS. So some of you may have seen this in reading. So if you see not, oh, that's a bad color. What's a good color? Black. NOS or INOS. That's nitric oxide synthase. And I is inducible nitric oxide synthase. These are the enzyme that convert arginine into this molecule, citrulline, with the release of nitric oxide. Okay, great. So that means if you want to cause smooth muscle relaxation, right? You want to make some nitric oxide. You want to do that. Okay, let's say you're having a heart attack. All right, you're having a heart attack. And I never know which one is which. So what is it when your heart is sort of beating irregularly like this?
AFib? No.
AFib?
Yeah.
Arrhythmia.
It's
Arrhythmia.
Arrhythmia.
Arrhythmia?
No,
AFib.
AFib is.
AFib
What's
is
the other choice then?
Are you talking about acetosinus?
When it's going like,
That's
when it's
AFib.
VFib, okay? Either one is bad, right? Both of them are bad.
But AFib is livable.
AFib is livable and VFib, don't you have to shock it?
Absolutely.
Okay, so, and what you're doing when you're shocking it is you're resetting all of the cells back to the same rhythm, right? So it's basically resetting all of, when you shock it, I think what it does is it opens all of the channels available, and so you're, basically all the cells are now going to have the same ionic concentration, and so that after the shock, it should just come back to a normal rhythm. It's a reset button, yeah. Well, and you know how that works on your computer sometimes, right? You do the reset and you get nothing. It's like, bam. So in this case, all right, they actually, one of the things they used to do is they had, what was it, pills? Little tiny pills, nitroglycerin pills, where they put it under your tongue, right? If you were feeling any sort of heart palpitations. And that was supposed to help you with smooth muscle contractions. What are you supposed to do? Are you still supposed to do that? Do
They
say
do
that?
it. It's a vasodilator.
Yeah, they do.
Right. I'm getting, so this is all about vasodilation. What is vasodilation?
Expanding the
Dilation.
So all of your arteries and your
Heart.
veins in your heart are surrounded by smooth muscle. And so when you squeeze those, that also causes problems, yes? So if you release the squeezing of the arteries, right, that's smooth muscle dilation. So the way that works is you have nerve terminals that release acetylcholine. Acetylcholine binds to a receptor. Inside the cell, that receptor turns on the nitric oxide synthase. Okay? And so these are endothelial cells. Where are endothelial cells found?
Lumen.
So that's what surrounds a vessel. Endothelial cells. And which side is the apical side in a vessel?
Inside.
In the lumen, right? The apical side is in the lumen of a vessel. So if I draw for you a blood vessel, which you know that's not going to be pretty. Here's a blood vessel, right? All of this is made out of cells, side by side, right? That are connected by junctions, et cetera, et cetera. And inside, right, this side is the blood is flowing. And then you have smooth muscle that's on the outside of this. So what this is doing is you're getting the activation of nitric oxide synthase, activating NOS. These guys are going to start making nitric oxide, which gets secreted. It actually just goes across the membrane, and it binds over here to something inside the cell, right? This nitric oxide actually got across the membrane, and inside here it binds to something called a guanylyl cyclase. A guanylyl cyclase. Oh God, that sounds painful, doesn't it? Guanylyl. Any word that has two Y's in it is not a good word. That's how I feel. Guanylyl. What is a guanylyl cyclase? Where do you know the word guanine? Guanine, thank you. So actually, this takes GTP, a guanylyl cyclase takes GTP and turns it into cyclic GMP. Ugh. What have we done so far? Let's open painful, yes? So this is a signaling cascade, right? So you had acetylcholine, AC, bound to a receptor. That caused this cell to activate iNOS. And this cell, right, so this is all one cell here, made nitric oxide. The nitric oxide got out of this cell, got into another cell, and in here it activated another protein called guanylyl cyclase. And that's going to end up making something called cyclic GMP. So what is cyclic GMP? Do you remember how to make nucleotides? O-P-O-P-O-P-O, O-P-O-P-O-P-O, with double bonds and all that shit. Do you remember this remotely? We could do it again. We could just have a little quiz. You're like, no. Right? So here was the G. Remember that? Let's make that a H and an OH. So this guanylyl cyclase, all it does is it takes this particular nucleotide and it converts it into a cyclic form, and it uses the energy associated with these bonds. What are these bonds? Those are the phosphoanhydride bonds. And it ends up, can I just erase and copy over this? Because what it ends up doing is you lose these guys. Oh, I can't, because I already moved it. All right. I need white. I'm going to white it out. Okay, so that third phosphate is going to end up, that's not pretty. So this oxygen had a phosphate on it, right? That first phosphate, right? And this is going to end up being attached to this oxygen down here. So you end up with, this is going to be CH2O, P, O, okay? So this particular molecule, right, is now a cyclic molecule. The phosphate is linked to the 3' hydroxyl. You lose that hydrogen, and you end up with one phosphate. What the hell good is that? So we're talking about you're going to make high levels of cyclic GMP. What was the point of this? So causing the smooth muscle to relax. This cyclic GMP at high levels causes smooth muscle to relax. Okay. So if we go back to this, right? Heart pain, angina, this whole process, right, can be done by activating this particular nitric oxide synthase. And it causes, they're showing you this smooth muscle right here is all bunched up. And then down here, notice, it looks so nice and relaxed. I'm just relaxed now. Nobody cares about this until I start talking about Viagra. Okay? This is how Viagra works. What does Viagra do for you? It does.
Good night.
It does do something for you. What would it do for you? It can reduce blood pressure. Do you think they were really looking for a drug that could cause a woody? They were not. Okay? After the fact, they were like, holy shit, this is a moneymaker. They were looking for a drug that could help with smooth muscle relaxation. And when they did this, they actually found that Viagra functioned very well in the tissue of the penis. Okay? For whatever reason, it seems to work really well. And so it allows for smooth muscle relaxation to be continued. Okay, this is a little bit of an oxymoron. Most people, when I say that, they're like, what? If you have a hard-on, it means that your smooth muscle is really relaxed. And what's that doing, Michael?
Blood flow.
Blood flow. It's all about blood flow. So you get increased blood flow, and that's what causes an erection. Right? If it's more than 4 hours, my brothers all tell me that they're emailing and telling all of their friends this. Because they're like, oh man. I'm like, don't call the doctor. Just go tell all your friends. Nut jobs. Gotta love having brothers. Okay? What does Viagra work on? Viagra works on, so this is a way better picture, yes?
Yes.
Aw,
Sorry.
come
It's
on.
much better than mine.
I did the best I could. So here's the cyclic GMP. Cyclic GMP is very short-lived in the body. It doesn't last very long. You have to continuously make it. And the way it gets degraded is through a phosphodiesterase. Ooh, that sounds like a phosphodiester bond, doesn't it?
Sure does.
It does, but in this case, it's a diesterase, so we're breaking a phosphodiesterase. So it breaks this bond right here. All right, and that leaves you with GMP. So that molecule can then go be recycled and be made back into GTP. But Viagra blocks this phosphodiesterase, and apparently it blocks the one that's in the penis way better than any other phosphodiesterase that you have in your body. Women can take Viagra, and it does lower blood pressure. All right? So that's why you should care about nitric oxide, not about Viagra. Okay, I know. None of you guys, you're not at that stage yet, but trust me, you'll get there. Okay. So there are the words for that. Okay, I gave you words. If we look at the hormones, so there's some time delay. All right. Going back to hormones. Okay. Cortisol, what's cortisol do for you? Stress. That's your stress response. Anybody feel stressed? Okay. I feel way less stress. Way, way less stress. As a matter of fact, somebody was looking at the pictures from the wedding. And like from before the wedding to after the wedding, it's like a clear distinction between me. It's like freaking out and like, it's the funniest thing. And I don't drink anymore, so it was just the stress. Okay. So cortisol, estradiol, testosterone, these all have that steroid structure, and they can cross membranes. What's this one down here? Do you remember? We did talk about this a little bit, yeah? Thyroxine, what's that good for? Thyroid. And what is this made out of? What's thyroxine made out of? What's that? Tyrosine. Exactly. It is. So it's made from tyrosine. So that's a good thing. Okay, hormones. So remember what we're doing. Okay, we're talking about the extracellular signals. The first one was nitric oxide. We're talking about a gas and how a gas signals. Now we're talking about hormones and how hormones signal. And some of the hormones get across membranes. Some of the hormones actually bind to receptors, like insulin. Insulin has to bind to a receptor. Right? So down here, what you're looking at is let me just make that part bigger. DHT, I wrote it in on the side, is testosterone. AR is androgen receptor. What's androgen? What's an androgen? It's an overarching name for hormones that are changing sex. It's about sex. Androgens are about sex. So in this case, testosterone gets into a cell and it binds to an androgen receptor. That androgen receptor is in the cytosol. When it binds, it causes, notice this androgen receptor is bound to all sorts of molecules. These are HSPs, or heat shock proteins. So there's HSP 90s and HSP 70s. There's a million HSPs, okay? What does the 90 and the 70 mean? No, that's a good idea. Those aren't numbers in order of being found. The size, the molecular weight. Okay, so some of them are bigger than others, so this is molecular weight. How do they determine whether they're going to name it on that versus the order they're found? I have no idea. It's just, I really think they're rolling the dice. All right. So once this binds to this androgen receptor, these guys get kicked off. So these guys help the androgen receptor stay in the cytosol. Once this binds, notice what happens. This androgen receptor gets phosphorylated. And this phosphorylated complex of testosterone with the androgen receptor binds to DNA. Okay? This binds to DNA and helps in transcription. What do you want to turn on when you, right? Think about what testosterone does. What does testosterone do for you? Anybody, if you take supplements, right, what do you take? You take testosterone because you want to be macho man, you want to build up muscle. So you're turning on transcription of things that are going to help you build muscle. Okay? Like when sex change operations occur, right? You need to change the hormones. You need to change the level of hormones. And women start taking testosterone to become more like a man. And what's the first thing that people notice in them? Hair, voice, and actually, sadly, I hate to tell you this, aggression. Okay? So they become extremely aggressive and not so nice. And I'm not saying that that's what men are. I'm just saying that I happen to know a lot of people that have done transition or at least seen a lot of documentaries on it. And so it's really interesting because their body's not used to that, and I think it's something going on. That's not what men are. Men are generally more aggressive, but they not like they're beating people up all the time. And so this is all about turning on gene transcription, right? Does that make sense to everybody? Somewhat? All right. So hormones. Just remember what we're talking about. We're talking about these extracellular signals and what they do downstream. It's just going to depend on what the cell needs at any one point in time. Insulin, right? Insulin does crazy shit. Like, you look at that and you say, really? What? You say lots of purple, and most people are going, you're shitting me, right? So, we talk about insulin, and I say to you, okay, the pancreas makes some insulin, it's secreted, that helps in skeletal muscle. It's a lot more signaling involved than just a simple putting GLUT4 on the plasma membrane. Okay, GLUT4 getting to the plasma membrane, so let's try and work our way back. Here is GLUT4. Do you remember what GLUT4 was? So it's the insulin-regulated glucose transporter. All right, and it sits inside a vesicle waiting, so here's the GLUT4, it's waiting for something to happen. That something to happen is insulin binds, and then somehow all of this happens. And it's regulated through RAB4. So RAB4 actually RAB4 holds it inside the cell, and when RAB4 gets activated, it comes off of this, allowing the vesicle to bind to the plasma membrane. And this is through PKB, which is protein kinase. You're not going to remember any of this, right? You're not going to remember this. What are you going to remember? You're going to remember that it's way more complicated than what I'm telling you. Okay? There's lots of steps involved because the cell is doing lots of things at the same time. All right. I don't think that's fine. So this is what, I mean, GLUT4 is actually in, this is a cell, I think this is a fat cell. Okay? You can see tons of GLUT4 intracellularly, and as soon as insulin binds, not all, but a lot of these are going to go and fuse with the plasma membrane, and then you get GLUT4 in the plasma membrane. Okay? That's pretty much what I want you to know. At me. Are you laughing at that, or are you laughing at this?
Yeah,
There's something in there? Okay. So this signal transduction cascade, right, is actually, insulin is the outside signal. It binds to a receptor. The receptor causes something to happen. So IRS is insulin responsive signaling protein. It binds to these two proteins, P85 and P110 are actually a lipid kinase. So they phosphorylate lipids in the plasma membrane. They make phosphatidylinositol, 3P. Then PDK is
It's written on the.
is it phosphodiesterase? PDK. Phosphoinositide dependent kinase. So once you get these lipids, this gets activated, which activates PKB, which is protein kinase B. Protein kinase B phosphorylates this protein, which interacts with the RAB, right? It's like dominoes. You catch on? Each domino hits another one. But we're showing you one of these at a time. Is it one?
No.
No, it's a billion of them at the same time. Okay? And all of this ends up causing this particular vesicle to break off of the intracellular store. Ooh, what's that?
A snare. That's anaerobic.
What's this? That's a cytoskeletal filament. So this is probably a microtubule, and this is going to be kinesin, because it's going to the plus end. That brings the vesicle to the plasma membrane. What's this? GLUT4. And now, Courtney, what's that?
A snare.
Snare. Okay? So all of this ties back to what we did at the very beginning of the class, yes?
Yes.
It'll all, right? The big light bulb should turn on. You should be like, oh, look at that. That's great. So there are the words for the previous slide. All right. So what have we talked about? We've talked about gases. We've talked about hormones. Okay. There are a billion other things that can signal. Ligands, and what do ligands bind to? Nicole, you tired?
No, I want to make sure we have enough time to finish the lecture.
We never have enough time to finish the lecture, right? I'm on slide 26 of 47.
Oh, yeah, we're not supposed
I don't think we're going to make it. Okay. So there are a ton of other ligands that can activate things. All right, and we're going to talk about some of them. Okay, I'm going to mention ion channels. So ion channels, there's different kinds of things. You can have a ligand that opens an ion channel. Okay, the ligand binds to the channel and it causes it to open. You can also have ion-linked ion channels. So an ion binds to a channel and it opens an ion channel. Oh, that's painful. We're not going to talk about this very much. We're going to talk about G-protein linked receptors and enzyme-linked receptors in gory detail. Okay? Because we can. Here's the ion-linked channels. This is showing you something binding. Here's an ion channel. It's gated ion channel. All this is facilitated diffusion. Right? It's facilitated by binding of a ligand. So when this red ligand shows up, it opens the channel and allows ions to go in or out based on what? Their concentration. It's just diffusion, all right? That's just a diffusion thing. They will go based on concentration. What do you need if they have to go against the concentration? You need a pump. So it's going to be some sort of ATPase because it's going to use ATP to pump against the gradient. Ion channels almost always are like this, okay? We've talked about cyclic GMP, all right? ions can also be considered second messengers. Why second messengers? What's the first messenger? So for cyclic GMP, what was the first messenger?
Acetylcholine.
It's hard to say, right? You could say acetylcholine, you could say nitric oxide. I don't know what they consider the first messenger, but cyclic GMP is the second messenger. Something happened before this, but this is the second thing that happened. All right? So, blah, don't worry. That was ion-linked channels. How do you like that? That was really simple. Okay, this is really not simple. This will be the last thing we do today. All right? G-protein coupled receptors. Why do I like G-protein coupled receptors? What's the G? It's going to be a GTP. So these are going to be GTPases. You remember GTPases?
On and off.
What's that? On and off? Yeah. What were the names of them?
Rab.
Rab, Rac, Ras, Ro, Ran, Arf, Sar, Dynam, blah, 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 blah. There's a billion of those. Those are considered small GTPases. These are considered large GTPases. They're in this class of proteins called G-protein coupled receptors. Okay, G-protein coupled receptors are 7-pass transmembrane proteins. Why do I care? The Kite-Doolittle plot, right? If you get a protein in a Kite-Doolittle plot that's a 7-pass transmembrane protein, you're virtually jumping up and down with joy, hoping that it is a GPCR, G-protein coupled receptor. Okay? Because we know so much about them, it would be great if that's what it was. So here's a receptor. It's going to bind a ligand. When it binds a ligand, it causes the recruitment of this particular protein. And this is a terrible picture of this. Terrible. All right? Because this has multiple subunits. The big idea is this binds a ligand, it recruits this to bind because it changed the conformation. Notice here it's straight, and once this ligand bound, it's curvy. How clever, yes? Okay. And then that caused the activation of something downstream. That's the big picture. The receptor itself, right, has this particular structure. Okay, it is called a serpentine receptor. Why?
Looks like a snake.
Looks like a snake. How clever. Now you guys, does anybody ever have to do the serpentine driving test? No, they don't make you do that. Here they're like, just go on the highway and if you can survive, you get a driver's license, right? That's pretty much like driving a serpentine. So they used to make you go around cones and make you not hit the cones, right?
Motorcycle test.
They do on a motorcycle test for sure. Okay, so serpentine, okay, is because it has this particular structure. This over here is actually, right, this is the G protein. And it's made up of 3 subunits. It's called a trimeric because it's made up of 3 subunits, alpha, beta, gamma. Alpha, beta, gamma. And the alpha is normally sitting in the inactive form with GDP bound. D, as in diphosphate. When the ligand binds, it changes the conformation of this intracellular loop. That then recruits this whole complex to come over. How is this complex linked to the plasma membrane? Lipids. A lipid modification. Okay. So where do I want to be? Let's just make this big. The words are on the one side, but if we look at this, this pretty much tells you what's going on. So when the ligand binds, it causes the recruitment of the G protein, and when this binds, what happens? It changes the conformation of the trimeric complex, and it kicks these 2 pieces off. So the alpha piece stays, the beta gamma subunits come off, and then what happens with this? It now has a totally different conformation because these guys aren't there, and it allows for GDP to come off and GTP to come on. Yay! And what do you know about GTP-bound GTPases? They do shit. Okay? They're turned on. All right? So now this is the active subunit, and they can do things. And there's different functions of different alpha subunits from GPCRs. We actually now know, so for a long time they thought this did nothing. the beta gamma, it has additional functions. I don't know what they are, but we now know they actually have functions. This is the 4th edition, and the 5th edition, I think it's the exact same picture. And so the alpha subunit has a function. Don't just cross this off. Just say fuck it. No, that's even better. Just cross. I don't care. Okay, blah, blah, blah. You do get, when you get rebinding of the beta gamma, okay, this is back when it's in GDP form. Here's the hard part of this, right? You're like, there's more hard part. Okay, there's different G proteins, different alpha subunits. So you can have an alpha subunit that's an S, you can have an alpha subunit that's an I, or an alpha subunit that's a Q. S is stimulatory. I is inhibitory. Q is totally a fucking weirdo. Okay? When I say stimulatory and G protein coupled receptors, it's all about making cyclic AMP. Okay, that's similar to what? Cyclic GMP. What was the enzyme that made cyclic GMP? Guanylyl cyclase. Okay? What's this enzyme going to be? Adenylyl cyclase. Yet another word that has 2 Y's. Fucking idiots. We'll pick up there. There we go. So G protein coupled receptors, right, have a, what does the actual receptor look like? Anybody? What is it? A snake. A snake. What's the other name we call those? Serpentine. Serpentine. It's a 7-pass transmembrane protein. That's the receptor. And what happens when the receptor binds ligand?
Activates the G protein.
How? 50% answer, dude.
It releases, I don't know.
50% answer in here?
Beta gamma come out.
Holy fuck, you don't know the 50% answer in here yet, people?
Standard confirmation.
You should have been doing your lab report. See, you missed it.
No, it wasn't.
Oh, you were. Come on, what the fuck were you doing? Is that not a lab
No, I was
report?
doing the roulette, but I was 30 seconds late. So
I'm not talking about today, I'm talking about the other day.
the other day I was paying attention.
You know what? You're not going to have any pants left because they have burned off, man. I got pants on fire. All right? All right. So it recruits the G protein, and what's the G protein made up of? Alpha, beta, gamma subunits. And it's the alpha subunit that we really talk about with respect to the activity. The beta gamma does have an activity. I'm not going to talk about it. It does actually do things. Now people have started to study that, and they're going to find different beta gammas that have functions. But we really know a lot about the G protein, the alpha subunit, that can either be GS, GI, or GQ. All right? And I said to you that the GS and the GI are all about generating or not generating cyclic AMP. The GS is stimulatory, so it's going to increase levels of cyclic AMP, and the GI is inhibitory with respect to cyclic AMP, so it decreases the levels of cyclic AMP. We talked about cyclic GMP with respect to what? Really?
Oh, smooth muscle
Smooth muscle
relaxation.
relaxation. Right? So cyclic GMP was important for smooth muscle relaxation. What was the enzyme that generated cyclic GMP? Right. It's right. It's got all those L's. Guanylyl. It's got 2 Y's, 2 L's. It's just a bad word. Guanylyl cyclase. This is done by a protein called adenylyl cyclase. And we're going to come back to GQ. GQ actually is a very different enzyme. It causes the activation of phospholipase C. So phospholipase is cleaving lipids, right? It cleaves a phospholipid. And this turns out to be really important. This one does. So this is just showing you how cyclic AMP is made, right? You have, okay, is this RNA or DNA? RNA. So it's interesting, most of the molecules that are used when we're talking about in the cytosol, they're usually the RNA form. And I didn't know that until recently. For whatever reason, I thought they were DNA forms, but like when we're talking about G proteins, RAB, RAP, RAS, RAN, RO, I think they bind the RNA form. They bind both, okay? But what's prevalent in the cytosol is this RNA form, so it has the hydroxyl there. When this gets acted upon by adenylyl cyclase, 2 phosphates come off and you end up with this cyclic phosphate group attached to the 3' hydroxyl. And phosphodiesterase comes in and cleaves where?
At the O.
Here, at this O. Here? No, here. How about here?
That's okay.
The bottom of.
Okay, it doesn't really matter whether you say it's here or here. This phosphate ends up, right? So you end up with AMP, adenylyl monophosphate. So this phosphate, and you end up with a hydroxyl there. All right. What can you do with AMP is just another messenger. Guess what it does? Changes, who said it? Who said it? What were you going to say? It binds to things and changes conformations of things. That's all we're doing. This whole course is just about binding and changing conformations. And all of signaling is going to be something happens that changes the conformation. That causes the function to change, so then it does this. So then it changes the conformation, and this thing does that. Little domino effect. I put these in here because I thought maybe it would help you remember it. So, cholera toxin. What's cholera? Bacteria that causes diarrhea. And is it prevalent?
Not here.
Not here. Thank you, Mo. Okay, so where is it prevalent? Third world countries where sanitation is an issue, right? So any place that really doesn't have running water, this is really easy to get into the water system, and people drink the water, and then all of a sudden everybody has diarrhea. This killed, I don't want to say millions because I'm not, it might have killed millions of people back in the day. It's very easy for this to show up. And what's interesting about it is the cholera toxin itself. So cholera is just a bacteria, it makes a toxin. The toxin keeps a G protein, that's a stimulatory G protein, in the GTP bound form. So it means it's continuously turned on. And that particular G protein, right, is going to cause there to be increased cyclic AMP around, and increased cyclic AMP in epithelial cells. All right, and so let's go back to Nicole. Nicole, you said it caused diarrhea. So what epithelial cells might we be talking about here? You're now Nicole. So you're Donna. Intestinal epithelial cells. In your intestinal epithelial cells, apparently what this does, the increased cyclic AMP causes there to be increased levels of chloride ion. And wherever you have lots of ion, what do? You start pulling water in because we don't like to have tons of ions anywhere. What does all that water do for you? Cleans you out. Right? So I did the colonoscopy last semester. That was some fun. And they make you drink this liquid, right? And the liquid that you drink is basically this salt solution. It's a potassium, it's got polyethylene glycol, which is interesting, so that's what's in antifreeze. I'm like, nice. Okay, and you have to drink a jug of it. Okay, it doesn't taste terrible, but it doesn't taste good. But after you drink the first couple glasses, you're waiting, you're like, okay. And so you're supposed to have like an 8-ounce glass every 15 minutes. Well, after 30 minutes, it begins. Right? So what happens is they're pumping your intestines filled with ions, and so all of a sudden, wham, okay, You're just cleaned out, man. You're cleaned out like there's no cleaning out. And just to make sure, they give you a laxative before you go to bed, so that when you get up in the morning, you're also like, oh, I gotta go. And there's really nothing in there. It's just water that comes out. I mean, clear water. It's amazing. It's like, wow, that thing's whistle clean. So, right? So if you don't want to go through all of that, just get a little cholera, right? And it'll do it for you. So Darren in my lab, he says that, people pay for, what do they call them? Colon cleanse. People pay money to have a colon cleanse. You don't have to pay money. Just look online. You can look up what the solution is. And really, if you do drink this salt water, it'll clean you out. If you're feeling bogged down, all right? So this is all about activating this GS protein. Here's another bacterial toxin, pertussis toxin. Has anybody ever heard of pertussis? Whooping cough, all right? Kids end up getting this, and it's a really bad whooping cough. That when they cough, it sounds like they're going hoo, ha, hoo, and it's like it really does sound like that. Okay, and the bacterial species is Bordetella. Does anybody else recognize Bordetella? From what? Dogs. What is it? What do you call it? What do we commonly call what they get? No. Kennel cough. So if you take your dog and you kennel them, let's say you're going away for the weekend and you take your dogs to the kennel, they almost always come home with kennel cough. And it takes a long time for that to go away. It's really irritating, and it's not fun for the dog. And they've gotten, it's not Bordetella pertussis, it's Bordetella, I can't remember. It's another Bordetella species, all right? And what's interesting is it actually works on a GI, G protein coupled receptor. And so this causes increases in cyclic AMP, and I don't actually know the mechanism of this one. So why that increase in cyclic AMP causes the coughing, but right, understanding how these work, right, would be a really great way to make what? Medications for them.
So the subunit is connected to the GDP, but it increases or decreases a cyclic AMP?
Correct, right? So we're just saying you have a protein, and when it's turned, this one, right? Mm-hmm. When this one's turned on, it activates an adenylyl cyclase.
Okay.
When this one's turned on, what do you think it activates?
The mono.
And it's not, right? It's not really turned on. It's actually, it keeps it turned off.
Right. Okay.
So you're not activating cyclic AMP.
Okay.
And this leads, so you're not activating a phosphodiesterase.
Mm-hmm. Not activating phosphodiesterase, which...
It's hard because this is an inhibitor of an inhibitor.
Okay.
Right? Nobody likes that. It's a double negative. Okay, so just understand that, right? We need to understand these functions because we can use these as targets in our thinking about making drugs. Cyclic AMP does a whole bunch of things, okay? I mean, it has multiple functions. One of the functions is to activate pKa. PK, protein kinase. All right, protein kinase A. Guess what? There's pKa, pKb, pKc. Oh God, okay? There's lots of protein kinases. This particular protein kinase sits in the cytosol. These are the kinase subunits. And they have bound to them these additional subunits. And when they're all bound together, the kinase is inactive. Cyclic AMP gets upregulated. And now cyclic AMP binds to these regulatory subunits. These guys come off. Now the kinase, protein kinase, is active. Nothing more than conformational changes. Cyclic AMP is a second messenger that's going to bind to things and cause activation or inactivation by changing the conformation. In this case, right, the regulatory subunits bind to cyclic AMP and come off, And now these guys can be active, meaning that they're good at things. Here's an example. So pKa, right, can actually be a really fast activation. So glycogen metabolism. Okay, what is glycogen, first off? Sugars that are stored, where do we store glycogen? In our livers, in our muscles as well, okay? We have glycogen stores. And they're basically these long polymers of sugars that are linked together by what kind of bond? An alpha-1,4 glycosidic linkage. All right? And the bear is coming to get you, and you're sleeping. That baby, man, that baby's killing you. It's okay? You're sleepy. Okay. Done. He's done. Just poke him with a fork. All right? So when the bear is coming to get you, and you don't have very much energy, what do you want to do? You want to immediately be able to mobilize some sugar so that you can make some ATP. Well, the fastest way to do it is to try and break down some glycogen. So now you have sugar. The sugars can go through glycolysis. You end up with pyruvate. It gets into your mitochondria, into acetyl-CoA, goes through the citric acid cycle, right? And so the idea is you want to try and generate as much ATP so that you can run away. So pKa actually is important for this glycogen metabolism. So pKa phosphorylates a protein that helps glycogen to be broken down. pKa can also have these really long-term effects, right? It can actually cause gene regulation. So breaking down sugar can be like this, right? As soon as you have sugar in your cell, it's being acted on. But some things, right, maybe you want to make more cells. Maybe you want to cause the cell to differentiate. And it can regulate those kinds of things too through binding to a cyclic AMP response element. It's a protein. Okay? Cyclic AMP binds to a protein. The protein now can bind to another protein. That can regulate transcription, and I'll show you how. This gets really complicated. They're not happy about it, but it's important. So this is showing you a picture of a G protein coupled receptor with a ligand bound. The conformation is changed. It recruits the G alpha subunit, and the G alpha subunit is already, right, got GTP bound. So we've already passed the point where it recruited, the receptor already recruited and let go of it. So it exchanged GDP, it took off the GDP, and the GTP is now bound. And what they're showing you is the GTP bound subunit, the alpha subunit, is activating an adenylyl cyclase. This makes the cyclic AMP, this binds to the pKa, releases the regulatory subunits, and now these guys, which are in the cytosol, can go to the nucleus. And in the nucleus, right, they're going to phosphorylate stuff. That's what kinases do. And in this case, okay, this is DNA, right? And they're saying that there are places in DNA that proteins bind. And one of these is called a Krebs binding element. So that means we're going to get the protein Krebs. This is the protein Krebs. No, I'm sorry, this is the protein Krebs. So Krebs gets phosphorylated. The phosphorylated Krebs then binds the Krebs binding protein. Why is it so complicated? Why do you need all of these things together? Why not just do one thing? Wouldn't that be easier? No?
It makes it a lot more specific. So like,
This allows for regulation at every single step. Okay? And by regulating it at every single step, you can have this high specificity that you don't have activation at inappropriate times. All right? And remember, this is not the only signal that's going on. This is just one signal that's happening in the cell at a time, but you probably have hundreds of signals that are going on. And there's going to be crosstalk from one signaling cascade to another. cyclic AMP, right, can bind to a whole bunch of things and cause different regulation of different events. So all of this ends up binding, and this is a transcription factor that turns on transcription of whatever the target gene is. Right? It's really complicated. Would you agree with that? I personally think it's really complicated. The GI protein is interesting because it's not doing anything with cyclic AMP, it's doing another whole signaling pathway. And this pathway is really important with respect to immunology. So what the GI protein does is...
GI or GQ?
I'm sorry, GQ. GQ. Thank you, GQ. So GQ is totally separate, and what it does is it takes this lipid, which is bound to the plasma membrane. So this is phosphatidylinositol 4,5-bisphosphate. What the fuck, right? What were the membrane lipids that we talked about like an eon ago? Do you remember any of them?
Phosphatidylcholine.
Phosphatidylcholine,
Phosphatidylserine.
phosphatidylserine. You're close. So there was PS, PI, PC. I think we were missing. So phosphatidylserine, phosphatidylethanolamine, phosphatidylcholine, and sphingomyelin. Do you remember those? There were 4 main phospholipids. So what the hell is this? What is PI? Another one. It's another phospholipid. Less than, I think it's 0.2% of your cell is phosphatidylinositols. But it signals. It's the only, it's not maybe the only, but for a long time we didn't think lipids had anything to do with signaling, right? We thought signaling was all about proteins, DNA would signal, but we didn't think lipids could do it. And now we know that's wrong. And phosphatidylinositol is this lipid that can cause signaling cascades. So this is a phosphatidylinositol, and this is the inositol ring. Right? All phospholipids have this particular structure, right? They have the 2 lipid fatty acid chains. They have this ring, this sequence up here, and then this is an inositol ring. And on this inositol ring, you can get phosphorylation on different positions of the ring. So this one, I think this is position 1, 2, 3. I'm not sure. Where's the O? So where's the 1, 2, 3, 4? What are they? This is 4, 5. So this has got to be 4, and this has got to be 5. 3, 2, 1. I did have it right. Mark it on the calendar. Right? 1, 2, 3, 4, 5. So this is a phosphate. This is a phosphate. This actually links it to the rest of the structure. But you can get phosphorylation on the 5 position, the 4 position, the 3 position. And any combination of those. All right? How is this going to get phosphorylated? How is a lipid going to get phosphorylated? What do you need to do that? A kinase. It's called a lipid kinase. In particular, this ends up being a phosphatidylinositol kinase. And there are particular kinases that will phosphorylate the 3 position. So if a PI3 kinase comes in, it's going to phosphorylate this position. If a PI4 kinase comes in, it phosphorylates this position. What if you have a PI3,5 phosphatidylinositol? Well, no, they're already phosphorylated. There's a kinase that will now phosphorylate the 4 position. So it's called a PI3,5 4 kinase. It's a fucked up system, okay? I'm not going to ask you to remember this because you're not going to remember this, but each of these has a different function. So we know that if you just have phosphatidylinositol, it has a function. If you have PI3P, it has a function. If you have PI5P, it has a different function. If you have PI4P, it has a different function. If you have 3,5, it has a different function. If you have 4,5, it has a different function. And there's even 3,4,5. Holy shit! So now there's all these different signaling molecules. Just to make your life obnoxious, yes?
Mm-hmm.
All right. So what does this have to do with GQ? GQ will cause the activation of a phospholipase. What do phospholipases do?
Break down the lipids.
They break down lipids. So they're actually going to cleave some of this off. So phospholipase C will actually cleave at this particular phosphate right here. And so now what it does is it releases this inositol ring that has 3 phosphates on it. What do we call that? That is IP3, inositol, right, 3 phosphates, IP3. And the molecule that's left in the membrane is it's still bound to a membrane. It's called diacylglycerol, or DAG. All of those molecules are then going to signal something else, right? They're going to bind to something and they're going to cause some change of conformation, blah, blah, blah. Are you? Yeah?
Trying to figure out what to memorize.
Try not to memorize, try to understand, okay? So in this image, the point of this, right, what is the point? Is you get activation of a receptor that ultimately downstream, right, somehow to go from this ligand out here to what the action is. The action here was to open a calcium channel, right, and release calcium out of the ER. What's calcium do?
It regulates.
Go back to the 50% answer.
Changes the conformation.
Causes conformational change, right, when it binds to things. I mean, I am not joking, people. All of your biology is regulated by these little things. It's a little tiny calcium molecule and it binds to a protein. It changes the conformation and can activate it or deactivate it. And it just depends what those proteins are. You're never going to know them all, right? If you work in a lab, if you are a doctor, right? If you're a dentist, you're going to specialize in maybe heart function, or like in dental work, maybe you're going to try and figure out how caries are caused by the formation of plaques. And all of it gets regulated by these little interactions. So how do you fix it, right? How do you understand? You can't fix it unless you understand how it works. So this process, I'm just going to go through, right? This recruited a GQ protein, right? A G protein couple receptor that had a GQ. Once this was activated by binding here, this GTP-bound GQ subunit caused the cleavage, right? So everybody loves cleavage. So it caused the cleavage of this molecule from the membrane. The release of IP3 caused the release of calcium, and that calcium caused something to happen downstream. All right? It's really, really hard to keep all of this in your brain. But when you work on something, right, that's what's, you want to be able to come back and say, I have some vague recollection of that, yes? All right? So G proteins had basically, if we just go back to what did G protein couple receptors do, right? They're all about adenylyl cyclase, and this was phospholipase C. So activating phospholipase C. PLC. There are other phospholipases. Phospholipases will cleave, and each phospholipase cleaves at a different place. You can't know all of this. Right? IP3 causes calcium. Calcium does all sorts of calcium-regulated things. And we've talked about molecules that are regulated by calcium. Anybody remember any?
Starts with C.
Starts with a C. Say a CA word. Calmodulin. Good one. It's right there. What other proteins are regulated by calcium that we talked about recently in junctions?
Cadherin.
Cadherin and catenins. Okay? There are a ton of these. Calcineurin is a really important one. It regulates immunological processes. You're going to see the further you go in your career, there's a ton of things that are regulated by calcium. Okay? I'm not going to talk too much about the diacylglycerol. It activates something called PKC. What's PKC? Protein kinase C. Right? What is protein PKA?
Protein kinase C.
So these things are going to phosphorylate shit downstream. The signaling goes on and on and on. All right? Yay, those are G protein coupled receptors.
Woo!
So what would you want to know from all that?
Everything.
Changing the conformation, and what are the things that start and what are the things that end? Like how can you end up if you have a GS protein? What are you going to end up with? More cyclic AMP, and what can cyclic AMP do?
Cause diarrhea.
Cause diarrhea. I love that. How? Think about diarrhea.
By causing
Influx of water. Why is water being dumped into your gut? Because there's ions there. Okay? It's as simple as that. How do we treat people with cholera? The best way. What?
You give them a solution of salts and sugars.
Well, you're trying to just keep them hydrated. The way people die from cholera is because of dehydration. It's as simple as that. We can give them antibiotics, but in third world countries they don't have antibiotics. But in third world countries they don't understand hydration. And unfortunately, this happens in kids, and kids are really sensitive to this, right? There's a point of no return. If you don't actually get enough hydration, it's really hard to come back from it. Okay, that was fun. Let's talk about enzyme-linked surface receptors. Woo, woo. These are probably my favorite ones of all time. Okay, does anybody recognize what's happening here? We've seen this molecule before. We've seen surface receptors like this that are brought together. Dimers. Things that dimerize. You know one of the dimerizes, Donna?
The
Herbs.
herbs.
Oh, Donna, you win the prize for today. You are my favorite person ever.
Today.
For about a minute. Okay. All right. So the herbs. They're also called herbs. H-E-R. Because heregulin is the ligand. Just in case you want to know. All right. So this is causing, there's some molecule that can cause these surface receptors to dimerize. Okay? It could be a single molecule. It could be dimer itself. It doesn't really matter. Once you get these receptors close enough together, what did they do? Do you have any recollection, Donna?
Other than RP2, they come onto them.
Right. What happens to the receptors, though? What's that red stuff that they're showing you? The Zila.
They give off to
Nope.
Emit
What's that red shit? No.
electrons are
They're not
excited.
No.
activated.
We're doing signaling. Mo? What? They're changing conformation. Okay, so you have two receptors here. They can't do anything because they're too far apart, so they get close enough together and they cross phosphorylate. Who said it? Angelica?
I did.
Good job. You're sick, huh?
Have allergies.
Yeah, good job. That's histamine in action. That's good stuff, man. That's snares. That's a beautiful thing, right?
You take Benadryl, so.
And what does Benadryl do?
Antihistamine.
Antihistamine. All it does is it mops up those histamines. The histamines are still being dumped out. Okay? All day. What are you allergic to?
I don't know.
I'm allergic to trees and grass. As it turns out, not such a good thing. Okay? All right. Receptors, all right, there's a ton of these. Receptor tyrosine kinases. Tyrosine kinase associated receptors. Receptor-like tyrosine phosphatases. What's a phosphatase? It takes off a phosphate. You have kinases, you have phosphatases. A receptor serine threonine kinase. Okay, we're getting technical now, right? You have tyrosine kinases? Why do we call it a tyrosine kinase?
It phosphorylates.
It phosphorylates what on the tyrosine? The hydroxyl group on the tyrosine. Remember back when I told you we had reasons that we have different sort of amino acids that are important? That's gonna, right? So what is the serine threonine? What do they have on them that gets phosphorylated? Hydroxyl group. How clever. Right? So this just means that you have a protein that can phosphorylate tyrosines, and you have a protein that can phosphorylate serines and threonines. And it's interesting, most of them are serine threonine kinases. Why can they do both? They're very similar. The structure of serine and threonine are virtually the same, if you have any recollection. Does anybody have any recollection of what serine looks like and what threonine looks like? Well, you know there's an OH there somewhere. Serine, what's serine? Are there two CH2s or just one?
Just
I think there's just one linked to an OH. And then the threonine, it's CH with a CH3 over there, right? And an OH.
Yes.
This is all supposed to come back together. Is it coming back together?
Yes.
I asked 107 students to draw for me two nucleotides bound together. I feel like I don't want to do this, but I might have to, right? It was a Moodle upload. I can guarantee that I could take those, look at those, and give them grades for the course from that one Moodle upload. I can tell who's going to get the A's, who's going to get B's, who's going to get C's, and who's going to get D's and F's from one Moodle upload, right? In 107, if you can't draw those, right, you're here, and some of you still can't draw those, yes? Anybody want to admit to that? No? Okay. I think the thing is that we don't pay enough attention to the structure, right? And these little things make a huge difference. So these guys, right, are all about the structure. Okay, look, a receptor guanylyl cyclase. Holy crap! So what's that going to make? Cyclic GMP. Oh God, a histidine kinase associated receptor. Fuck. So, yeah, so it's actually a kinase, right? That's bound to a histidine on a receptor. And what do you know about histidine? It sucks. So, is it charged?
C.
C. C means yes? C. C means average. So, it's charged. What charge? Positive. What's its pKa? It's the only one that you really should actually have any clue what the pKa is. 6.5. So it's the only one that actually changes a lot around physiological pH. Right? All the other charged ones change only at really high pH or really low pH. Histidine's the one that's really going to play a role at physiological pH. So there's a billion of these receptor tyrosine kinases. Okay? And we know now that tyrosine kinases are the #1 receptor that's got problems in cancer. So RTKs, receptor tyrosine kinases, if you mess up their function, cancer happens. And it's just really easy to understand this because a lot of them are growth factor receptors. Okay, so if you mess up growth, what do you end up with? Overgrowth, cancer. Okay? So these are showing you, right, you can have different domains on the outside of receptors, but they all have some sort of kinase domain on the inside. It's funny because these F receptors, right, EPH, F receptors, are the most numerous, and they're the one I know the least about. Okay, they're important in cell adhesion and migration. But all of these, right, what's NGF?
And growth factor.
N growth factor, good. What's EGF?
E growth factor.
What's E?
Epithelial.
Epithelial? Neural. PDGF is platelet-derived growth factor. Okay, so that has to do with blood cells. VEGF? Vascular endothelial. Vascular endothelial. Why do we care about vascular endothelial cells?
They're pretty important.
What do they do? What is it? What is the vascular endothelium?
Vessels.
It's all about vessels, right? You're making the vessels. And so when you mess up vascular endothelial growth factor receptors, okay, it's all about giving tumors blood supply. Oh, darn, right? If you can cut off the blood supply to some tumors, right, you can kill them. But we really can't. We really can't do that very well because it turns out you need those blood supply for you. Oh, darn. These receptor tyrosine kinases kind of work the same way. They're too far apart. When they bind ligand, they get close enough together, and that close enough together then allows one chain to phosphorylate the other chain. That's called cross-phosphorylation. Okay, once you cross-phosphorylate, right, now I'm all phosphorylated. What's happened to my chain? Changed this conformation, and now I can auto-phosphorylate, right? It's like, you know, you're sitting on the couch and you get up and move to another room, right? Then you can do as much you want to yourself as you want. Okay? Somebody had to actually get you to get up from the couch, right? Your brain had to say, okay, I want to do, never mind. I'm just in the gutter. Okay? Autophosphorylation is, right, just tickling yourself so that you now have a different conformation. And what is all that going to do? Change your function. Does everybody know this word, oligomerization?
Many polymerase.
It's just putting a bunch of things together. So if you have a monomer and you put a bunch of monomers together, that's oligomerizing. Oligomerization is the process of doing that. Polymerization. What's polymerization? Making things. Okay, so this is the classic, this could be epidermal growth factor receptor, EGF receptor. What's the ligand if it's EGF receptor? EGF, you were right, Courtney. Don't just give up on yourself, okay? So if this is EGF receptor, the ligand is EGF. Okay, so we have another name for those. What do we call them? Herbs. They get close enough together, they cross-phosphorylate, then they auto-phosphorylate, and then they do all of this activation downstream. Oh my God! Look what one of them is. Phospholipase C. We just did phospholipase C. What does phospholipase C do? Cleaves what? Phosphatidylinositols, right? And makes the IP3 and DAG.
This is the other half of that same picture.
I know. You're so right. Okay? So there's going to be crosstalk, right? So some receptors are going to do the same thing that another receptor does. But then there's some things it does differently. And the two that we're going to talk about in gory detail is the MAP kinase pathway. And look, PI3 kinase. Holy crap! What's a PI3 kinase do? Mo? Now you're Mo.
I can be a lot.
I see that. You're morphing. So Donna is now Mo, and Mo says that we can phosphorylate the 3' position of phosphatidylinositol. Oh, that's a membrane lipid. It's not just in the membrane. It can be in any membrane. Okay? All right. So this is what we're working towards. This is what you are working towards understanding. Okay? Your facial expression says to me, you're so unhappy. Am I right?
I'm lost.
You're lost? Well, let's get you back. Tell me why you're lost. You're not the only one. How many people are lost? Okay? It's not like I don't know that you're lost, right? You're feeling stress. Am I right? Because this is a lot of information. Are you okay? Should I go talk to someone else? That's okay. So, who else is lost? And tell me why.
It's just a lot of info, a lot of words that all sound the same.
It's a lot of words that are new, and maybe it's just you feel like you're not going to get it. And you're going to get confused, and then you're going to fuck it all up on an exam. Right?
Exactly.
I get it. I'm with you. I totally understand that. So, I'll tell you how to try and get this. Right? You take one pathway at a time, and you say, okay, what's the big picture? Why do I care about G-protein coupled receptors? And sort of go down that pathway. What can G-protein coupled receptors do? And you know that there's 3 G-alphas. So you have G-alpha, that's going to do more cyclic AMP. Right? And you just try to work your way down one pathway at a time. And I really recommend drawing the pathway out as you go. Right? The only way you're going to get this is by practicing. If you just try to memorize, okay, just with words, it won't work. All right? You can't even make enough, what are those things that people make, like little, no, like little acronym things. Mnemonics. There's not enough mnemonics to cover this. Okay? Because then you're just going to mess up your mnemonics. You're going to get them all mixed up. So just try one thing at a time and see how it goes. Okay? You should feel overwhelmed. So how many of you are right where you should be? Right? That's good. So you are overwhelmed, and that's what you should be feeling. I'm sorry. I can't help it. Let's go to the next lecture, and let's walk through another one of these. Okay?
Did you get all this your first time learning that?
So it turns out that I'm an idiot savant about stuff like this. Was that you, Michael, that asked this? Yeah, so I seem to have this